think the way I became interested in Alzheimer's research really shows about how the field's progressed over the last 30 years. I have an original training as a marine biologist and did additional training in cell biology. And when I was looking for a faculty position, I was approached about applying for a job to study Alzheimer's disease. I didn't know what Alzheimer's disease was, and I was working at a medical school, Baylor College of Medicine at the time. I asked my colleagues there, and none of them really knew what it was. So I interviewed for the job and saw that there was a lot of potential because it was really the ground floor of the disease. At that point in time, in comparison, there probably were 50 papers per year published in Alzheimer's disease. Now there are over 5,000 to 10,000 papers per year. So it really was an opportunity to apply cell biology and even marine biology concepts. Even though the brain is quite different than marine organisms I studied when I studied marine biology, I applied the same techniques. The center part of my work is in oxidative stress. We know a lot more about Alzheimer's disease than we did 30 years ago, but we're still studying the basic things that came about when Alice Alzheimer described the disease in 1906, the plaques and tangles and how they fit into the disease. And we made major headway in diagnosis of the disease, the early detection, biomarkers, and in the last few years, some of the biggest headway has been understanding lifestyle, how diet, exercise, and many of the other things that people do make a difference in the disease. And therapeutically, we've made some minor inroads, and I can talk more about that later. But in terms of a cure, an effective cure, we've not made very much headway. I think we're on the verge of making major progress because we understand so much of the foundation of the disease, even if we don't understand all of its underpinnings. You know, most of the things that protect you from Alzheimer's disease would be the type of advice you would get from your grandmother. Live a life in relative moderation, you know, exercise, eat a balanced diet, maintain happiness, you know, the things that people are being guided to all the time, eating less meat, lower carbohydrates, those type of changes. And those are revealing themselves initially by differences in incidence of the disease among different groups. Initial work 20 or 30 years ago was showing people with lower education or people that had some poor diets, etc., might have an effect. But now it's becoming more obvious. Ethnic differences, people, African Americans and Hispanics have 2 to 3 times higher incidence of the disease. It isn't clear completely why, but it's probably in part genetically related, but more likely lifestyle related. Women are more affected, and since women live longer, they're even more affected. I used to take care of a brain bank that collected brains from people with Alzheimer's disease, and from that I had the impression from the brains that we collected and donated to our program that it was half male and female, and that most people affected were in their 70s. Neither of those are the case. The reality in the population is most people with Alzheimer's disease are female and are much older. So the classic Alzheimer person really in the population is an 80-plus woman. There's been a lot of attention about the vaccine for the last approximately 15 years, and the vaccine has succeeded in its first goal. It's removed the pathology of the disease. The initial focus was on removing amyloid. Subsequent work was on removing tau, so both plaques and tangles would be removed. Both of those are still ongoing studies, but there was no benefit to patients. In fact, patients got worse. There are 4 approved drugs. 3 are cholinesterase inhibitors, and those drugs are based on the cholinergic hypothesis of the disease, which developed during the late 1970s, early 80s. And that is that there are cells in the brain that produce acetylcholine that die, And the idea was that you could die during Alzheimer's disease, that those drugs make the brain more effective with the remaining cells. They're based on the success that was done with Parkinson's disease with L-DOPA, because the dopaminergic neurons die, and if you give people L-DOPA, there's a clinical benefit, at least for some time. They weren't as effective in Alzheimer's disease, but they do have benefit for patients. Most people claim that they have benefit for 6 months to a year for about half, at most half the patients. Some people think that they actually have benefit much longer than that. There are some people who think that you shouldn't stop it. It would also depend whether you have a lot of side effects from it, gastrointestinal problems, and very often people move from one drug to another to see if it has different portfolio. Those are the cholinergic drugs. And since you don't really benefit from taking the drug, what changes is your rate of decline. It's not so obvious how to spot these changes. If people take the drug, they might get a little bit better for a short while, but more likely what is that they would just not decline as much. And the benefits that have been claimed for longer use are more in stabilization of behavior than memory issues. The major issue for most patients that have Alzheimer's disease is not memory loss. It's behavioral issues. The other drug, Namenda, works by a different mechanism and usually used in conjunction with the cholinesterase inhibitors. One of the most exciting things I'm working on is trying to exploit something we've been working on for over a decade, and that is cellular models of Alzheimer's disease. What's hindered progress in Alzheimer's disease has been inappropriate models to study it. Animal models that are genetically modified mice that have mutated proteins in them have not been very predictive in therapeutic studies. In other words, these mice have been cured hundreds of times by different drugs, and you apply those same drugs to people and you find no benefit because the mice don't normally develop these diseases. But there's now new technology to be able to take cells out of people that have Alzheimer's disease and modify them to be very similar to brain cells. Because the problem of studying brain, as I usually study human brain, is most of my samples come from dead people. And I can observe them at various times during the course of the disease, but I can't manipulate them. And having these cells derived from people's brain, and we've done studies from fibroblasts, skin cells in the past, and we know that people have an Alzheimer's-like change throughout the whole body. The same changes you find in brain, you can find in skin cells and blood cells. But the problem with that is that the skin cells and blood cells are not neurons. Neurons are very unique. They're very large cells. Some of the cells in our body that are neurons are 3 feet long. Now, the ones in the brain that are affected with Alzheimer's disease aren't 3 feet long, but they could be half an inch long. And to be able to study cells that have such long extensions, axons and dendrites, offers an opportunity to really get at the disease. Fundamentally, Alzheimer's disease is a disease of disconnection of how the brain functions. The main thing about the brain is you have all these little transistors called neurons, and they connect to each other with thousands of little wires called axons and dendrites. Those wires don't function well in Alzheimer's disease, even before the neurons die. What I'd like to look for is to really understand the cellular and physiological basis of Alzheimer's disease. And I'd like to think it's going to happen next month, but it's more likely to happen over years of study. Having a cellular model where you can really get at the intrinsic changes of the disease, I think is going to advance things substantially. And further, in terms of a therapeutic, which is what I know most of our viewers would really care the most about, this type of cell model also, in addition, whether we get a complete understanding of the disease, will be important for screening compounds. In other words, looking at drugs that may be able to restore them. Most drugs that have been developed are developed for diseases that are not completely understood. So we may even be able to develop something that's efficacious that will not require us to completely understand the fundamental piece. The other part I study is the oxidative phenomena. That's the thing I'm best known for, understanding oxidative stress and free radicals. And why do people care about that? Well, they focus on taking vitamin E and vitamin C and eating blueberries because they care about that. But it's really a very complicated effect. It involves metals, iron and copper. It involves mitochondria. And we're really trying to dissect that. And I'm doing experiments along those lines at UTSA. One of the things I'm studying with a high-resolution electron microscope is how copper and iron are organized in plaques, and making important findings showing that the copper is associated with plaques and protein. On all my studies at UTSA, I'm working with about 10 or 15 people, and throughout the world, hundreds. It depends on the type of drug, but a standard drug that starts from the beginning, probably about 15 years. Because the first part, if you start from a drug that has never been used before, you'd have to, and you had indications for it, you would then do safety studies, phase 1. So you would use some animals first, and then you would go to people, normal volunteers, and you check that the drug didn't cause problems. Then, if that all checked out, then you would go to phase 2, which is a small clinical study with a group of patients, mainly to look for dosing. That would take probably a couple years, because the way Alzheimer's disease is defined now for clinical benefit, it means that you're looking for a lack of decline. All things could change if the patient suddenly miraculously got better. But that is not the paradigm that's been defined for the disease. So if you're looking at lack of decline, you have to go about 18 months. You also have to collect a fairly large number of patients, even for phase 2. And when that pans out that there's a signal, in other words, some patients appeared to benefit by taking the drug more than the ones who didn't, and of course, again, it was still safe, then you would have a large phase 3 study, which might be sufficient to be able to prove efficacy. That requires raising from all the way phase 1 to phase 3, probably $60 to $100 million at least. And then there's still more steps, because sometimes you have to do another trial, even larger. So by the time you put all of that together, you're looking at just the test, and if everything did well, probably about 7 or 8 years. And more likely it wouldn't go that fast. That's why I was saying more the 10 to 15 year range. From a research standpoint in what I do, early detection is really critical, because I think one of the big things now in Alzheimer's disease is understanding this transition of normal aging into Alzheimer's disease, and understanding if there is a pre-Alzheimer's phenotype, and is that really Alzheimer's disease or normal aging? And that piece needs to be dissected. There's a lot of interest in saying that Alzheimer's disease begins 20 years before the clinical aspect. Based on data we've obtained, I don't think that's the case. I think it's just that you're looking at the transition between aging, which means you're more prone to having many diseases, and having Alzheimer's disease. My concern, at the same time, I think it's really important for the work I do. My concern about early diagnosis for families, extremely early diagnosis, and by that I mean pre-symptomatic diagnosis, is it may lead to a lot of unnecessary concern. Number one, not everybody that has pre-Alzheimer-like changes converts to Alzheimer's disease. If all of us live long enough, our chance of having the disease is extremely high. If we live to 85, our chance is probably 30 to 50%. I just think it creates more worry than necessary. Now, that would all change if there was a better therapeutic. If there was a therapeutic that made a difference during that pre-symptomatic phase, then earlier diagnosis would be useful. But that doesn't exist now. I think who should administer them depends on the environment you're living in. I mean, all of us, I would like to have administered by the world's expert in being able to do neuropsychological tests and imaging and biomarkers. And that may exist where you are. If it doesn't, there are a lot of other practitioners that can be of value. What's been shown to be the case, even though it may not be the most sophisticated, neuropsychological tests that administered by people that are skilled are incredibly sensitive to spot Alzheimer's disease. Now, those can be buttressed by new biomarker tests. Those are not being used in the standard fashion. Both blood tests, cerebrospinal fluid tests, as well as imaging tests. Right now, the gold standard for Alzheimer's disease still is neuropsychological testing, because Alzheimer's disease is a clinical disease. It is a disease of loss of cognition, dementia, loss of function. So if your definition is that, neuropsychological tests are going to be the most valid and administered by people that are quite familiar with it. My largest supporter over the years has been the National Institutes of Health, the Alzheimer's Association, and then some other Alzheimer's. But one of the things I'm most excited about is being supported by the SIMS Foundation, because having unrestricted funds, in fact, was really essential to being able to work collaboratively across the world. Use those funds to support a student at UTSA, to bring a postdoc student, well, she's both. She was my student and postdoc from Portugal to visit for a week. And I had one of our postdocs that works with me at UTSA be able to go to a collaborating lab to prepare the senile plaques that being used for electron microscopy. There's a huge need for funding. The government is continuing to put more money into Alzheimer's disease. Unfortunately, a lot of it has been directed to the vaccine or amyloid removal, which has not borne a lot of fruit. But that approach of understanding how amyloid fits into the disease is critically important. It's Alzheimer's disease, just as it's defined clinically by the dementia, in terms of biologically and pathologically, it's defined by plaques and tangles. So understanding how they fit into the disease is critical. Well, the official line from the Alzheimer's Association is that working for the cure. Cure means reversal. Some of the work that we're doing with a company, Neurotest, which I'm a chief scientific advisor for, is looking at changing the metabolism of people early in the disease to reverse it. But we haven't succeeded. We haven't even done a clinical trial yet. We've only done animal studies. Many people are trying to figure out a way to reverse the disease. But, you know, to have a benefit for real patients that are living, just to be able to reduce the disease and delay it by 5 years will make Alzheimer's disease 50% less in our population. Just a 5-year slowing of the disease.
It would be a huge financial savings.
Financial and 5 more quality years for family. So if you ask what would I ultimately like to do, of course I'd like to end Alzheimer's disease. But, you know, if we look at other diseases that are common in the population, age-related heart disease or others, we don't really cure them. We do things to make people live longer, have a longer life. I think that's a more realistic goal. And then from there, you can move to the cure. Very little. My work has been pathological and biochemical. Most of the Alzheimer's patients I've encountered have been ones that I've removed the brain from at autopsy, and I don't deal with them that much in living state. Extremely so. People with Alzheimer's disease and family groups are incredibly generous to move forward. In fact, almost every brain bank in the United States has an abundance of brains because of that. And that's not true for every other disease. Yes, and brain donation, you know, it sounds like if you have a huge number of brains in a brain bank, and the brain bank I work with the most often, we have close to 400 brains. So then why do we need more brains when most of my studies only use 10 or 15? Things change. Brains, how they're collected and how people are analyzed clinically changes through time. When we were doing those studies 30 years ago, we still have brains from when we collected them 30 years ago. We were interested in people with late-stage Alzheimer's disease to isolate plaques and tangles, and we're still interested in that. But if you start looking at the transition of normal people to Alzheimer's disease, you need to have exceptional clinical data. You also need to have people that are incipient cases, the people that are just developing the disease. And those are quite difficult to collect. Well, from my own standpoint as a researcher, Alzheimer's disease, when I didn't know anything about it 30 years ago, and from what I have learned over the last 30 years, I think it's a miraculous disease to understand the aging process of our most complicated part of our body, our brain. And I've actually come to an appreciation of amazement that not that people have Alzheimer's disease, but that most people, the majority of people, live a long life and have normal cognition. And they don't do it just by having beautiful brains. The brain is constantly rewiring and modifying itself so that we function. And understanding that and understanding Alzheimer's disease, I think it's going to give us a tremendous insight into ourselves. You know, if you'd asked me that question 2 weeks ago, I would have answered clearly it has benefit. I know in some recent studies there were some indications that didn't have benefit. What I can say, because after these studies I'm less clear that it has a direct benefit, doing things to keep you mentally and socially involved are really critical. If I was talking to a group of donors, the UTSA, I say giving to UTSA. Whether you're giving to UTSA or you're volunteering in a food kitchen or helping the aged or doing anything that keeps you socially and actively involved. Now, if you don't want to interact with people and you can only do that through games, well, that's an alternative. But it's very important to maintain a positive outlook. You know, the aging process certainly is one of generally decrement. But there's a big difference between those people that maintain a positive attitude during that decrement and how their decline is. One of the most important new connections in Alzheimer's disease, and I'm sure it impinges on lifestyle, is depression. The people with one of the major predictors of conversion of an old person to having Alzheimer's disease is depression, and people that have Alzheimer's disease very often are depressed. And groups that you think are very happy or have anecdotally, like Hispanics, actually have extremely high depression levels. So there's a lot of things that we need to understand, this lifestyle and what you can do to keep a positive attitude. You know, I cannot answer that question because the problem is many of the antidepressants have a lot of side effects. So I think you have to do it with special care. It certainly would be useful to go to somebody who's very skilled, a neuropsychogeriatrician or somebody that has that skill set, because you don't want to over-medicate people. The issues of behavioral issues for older people, as well as especially those who have Alzheimer's disease, are major issues for families, because people with Alzheimer's disease have depression issues, they have aggression issues, wandering, many of these things. And the pharmaceutical industry is not directed their efforts at. Most of the efforts in Alzheimer's therapeutics have been on memory, which is important, but it isn't the major problem that families have to deal with. You can forget about a lot of things, and the family can deal with you. But if you have aggression or wandering, the family may not be able to deal with the issues very well. That's one of the newest things in neuroscience over the last 15, 20 years. There was a major change in our understanding of the brain. But before then, it was felt that the brain was developed in early development, you know, and during childhood, and all the neurons you'll ever have were developed then. And what's been shown to be the case is that neurons actually arrive all the time during your life. There's stem cells actually within the brain that form neurons. And then by understanding that, understanding how could you get more to turn into neurons. And then it was found to be different factors. One of the biggest factors is exercise. In fact, it's said that exercise would have more effect on you being able to learn something than anything else.
And is that like aerobic exercise? What type of exercise, if somebody were wanting to know, what's the best thing to do?
You know, I think I don't know the answer to that if it's aerobic exercise. I think it's moderate exercise. I don't know if you're benefited by running a marathon. You know, there are, for most of these things, studies indicate that there's a value, and then you actually hit a high point, and then you actually have negative effects once you get to higher levels.
So get out and take a good brisk walk.
A good brisk walk.
What does that mean?
Well, this is a major implication of looking at Alzheimer's disease is systemic, and certainly we have strong evidence, and other people have found the same, that all the cells in the body have changes that are similar to the brain, not the same. And if you can model that outside the body on a dish, it means you can have Alzheimer in a dish, because you would be studying the disease that was actually present within a patient, not something you created. Because when you genetically modify, like mice that are mostly studied for Alzheimer's disease, that disease doesn't exist in their biology. It's where these cells have this change in their biology. We've already made, at UTSA, the precursors of these cells. We've taken skin cells and we've de-differentiated them by the technology that was just awarded the Nobel Prize. It's where you insert 4 genes into a cell, and that cell then becomes back to a de-differentiated cell. So you take a skin cell and you put these genes in, it becomes kind of a amorphous type cell, and that's the precursor cell of forming any of the cells of the body. We're particularly interested in neurons. As I said earlier, neurons have these long projections that they use to connect to each other, and that's what we'd like to study, how they're modified, and particularly how the mitochondria, which are the energy producing parts of cells, we know they're different in the skin cells. We haven't documented yet whether they're different within the cells after we've modified them in this way. All of those tests are valid. The clock test has a big advantage because this is just simply asking a person that might be suspected of having Alzheimer's disease, or screening anybody over the age of 50 might take this. They just draw a clock and put the hands on the clock at the time specified by the tester. Those tests, like the clock test, should be followed up with more detailed tests. The mini mental test is also a quick test. It involves asking questions of patients to recall various facts of life or numbers, etc. And there's various degrees of sophistication for these tests. If they're administered at an appropriate battery, like the ADAS-COG, and I'm using technical terms there, more detailed ones, they have a huge amount of predictive value to define Alzheimer's disease. Why are the tests important further, not just for early detection? It's important to know if somebody really has Alzheimer's disease or the other conditions that masquerade as Alzheimer's disease. Because depression could be just depression, not a precursor to Alzheimer's disease, which could be treated. And there are other conditions such as frontal temporal dementia, Pick disease, diffuse Lewy body disease, and those have had increasing attention. Robin Williams was diagnosed with diffuse Lewy body disease. Some other famous stars have progressive supranuclear palsy, as where there's undoubtedly more. It was felt to be an alcoholic, which is what he played in movies, but I don't know if he was an alcoholic or not. But the reality is he had progressive supranuclear palsy, which is another debilitating disease. All of these diseases can be diagnosed by neuropsychological testing, together with a neurological evaluation by a neurologist. I know people are concerned when I talk about clinical trials that might be 60, 100 million, if not more. In fact, final approval of a drug very often costs $1 billion by the time you have all the marketing and all the other pieces. So then if you think of any sum you would give, wow, how could that make a difference? The answer is it does, because the type of work I'm involved in and most scientists are involved in are not clinical trials. We provide the foundation for those trials, but those are undertaken by companies. They raise money in the private sector from investors, and if you want to contribute in that way, I can even give you the names of some people to invest in. But in terms of basic science research that brings the ideas that will eventually go to clinical trials, much smaller investments. Investments of hundreds of dollars make a difference for reagents, for attending meetings, for paying for students. Most of the research is done by students and young fellows. I was one of those one time. I did all of my own experiments. But at this point, most of the time I work through other people, and that's true throughout the world. And those people are not earning huge salaries, not millions of dollars salaries. They're very dedicated, and they're getting trained, and they'll be the future workforce for Alzheimer's disease. Over my lifetime, I've impacted and had trainees, about 50 different people have studied with me, and those students are still involved in Alzheimer's disease throughout the world.